So for today's meeting, we're going to have two presenters. Tristan's first going to talk about and show us some things that he saw at the FENS conference last week. And after that, Vivian is going to continue the discussion about grid cells with some more literature review. And I think without further ado, I hand it over to Tristan.
Okay, howdy. Let's see. Share my presentation. You should see my presentation, yes?
Yeah. Well,
Okay,
yeah.
cool. There we go. All right, so hi. Hi everybody. I'm Tristan Slaminski. I was I attended the FENS forum last week. Uh, this is a big event. Just neuroscience. There's like 8,000 neuroscientists attending. There's like thousands of abstracts. So there's lots and lots and lots that is happening there. Big gathering of neuroscientists in Europe. So this will be just a few selections that I stumbled upon that I found are related to Thousand Brains project. Some of them might be more related than others. Some of them might be tangentially related. So this will be, I have about 7 topics to highlight. And if you have questions, I will be happy to tell you to talk to the authors of these things. I don't know how much I will be able to uh, explain beyond what I heard, and I will do my best to explain why I think it might be relevant, or at least what I thought uh, was what compelled me to highlight these things. So, 7 topics to get through, so let's just get started. One of the interesting highlights I thought was that there is a fixed number of grid-like modules emerge regardless of scale. So this stood out to me. It comes from FeetLab from MIT, uh, who, who was co-authored papers with previous Numenta folks about grid cells. This is what, regardless of scale, was the interesting part, and about there is a mechanism highlighted here that shows how computationally how grid cell-like things can emerge. So I thought, since we care about grid cells being in some kind of way inside the neocortex cortical columns, I thought this was interesting. More specifically, uh, they have a computational model where using using where it doesn't matter how much space you have, the same fixed number of grid cell modules arises from two simple properties. So basically, on the bottom it says modules scale automatically with size with variation in brain size. There's no change required in lateral interactions. So I'll talk about more here. Here's the
Hey,
principles. Here's how it
Tristan.
emerges. Yeah.
Tristan, I'm sorry, confused already.
Yep.
Um, what do we? It says emerges regardless of scale. What scale are we talking about here? The size of the brain, the size of the environment that the animal's in, the size of the scale of the grid cell modules. I'm missing what's the headline referring to?
Um, yes. So the scale, as in
The size of the neural tissue, as far as
the
I
size of the
understand,
neural
like
tissue.
how
So you
many
can
Right.
have
neurons is
small
available.
neural tissue like a cortical column size, or you can have it wider, like
So would this another way of putting this is that all brains have the same number of grid cell modules? Is that one way to put it? Like big brains, small brains.
um,
Uh,
this is a computational simulation result. They just have a mechanism for if
okay.
you vary two parameters, no matter how much space you can expand this model in, you will still get the same number of
Okay.
modules.
Got
But
it. So I
but
mean, yeah, go
but yeah, I
ahead,
think in
Vivian.
the paper they talk about that this kind of explains how you can get grid cell models in different species with different brain sizes without having to
Okay.
do a
So
lot of
it's
genetic
it's a
variance
proposed
there.
mechanism that explains the number of grid cell modules and their relative scale that would emerge through some mechanism that they're proposing. Is that would be a good summary then?
Yes, I think so. And it
Because
shows how,
I've never heard anyone suggesting there's different numbers of grid cell modules in different animals. So to say there's a fixed number is okay. That seems to be always the case. All right, but this is really a mechanism, right? You can say, here's how this could come about, something like
yes,
that.
and how it
Okay.
comes about from two smoothly varying things, you get
Hmm.
discrete cutoffs in modules.
Okay. Thank you.
Um, so here are the principles that local interactions without modular structures can give rise to globally modular grid cells. Uh, the entire process relies on two local interactions. One is in fixed scale, one smoothly graded. I'll talk about that shortly. And then peak selection on roughly energy landscape as general mechanism for modularity emergence from neurons to ecology. We're going to skip that. So this is the mechanism. What this figure shows you is up top in the green and the yellow. Oh, I have two mice. That's interesting. Okay, there we go. Green and the yellow. Um, the green represents one local relationship where it's like smoothly varying the width of the Mexican hat activation varies smoothly across the one-dimensional space here. So on the left here, you have a very wide kind of Mexican hat activation, and then it gets narrower as you go to the right. And then the other local interaction is this sort of fixed distance interaction. So on the left, it's the same. The orange distance is the same. It's the same in the center and the same in the left. And so it just turns out that if you combine these two interactions together, what you will get is when you get the peak activation, the third thing that they talked about is when you combine these two simple local mechanisms, no matter how wide your one dimension is, you will get a fixed number of these modules that are kind of correlated in how they respond to different inputs. So you'll get a module one, module two, module three, module four, and if your smooth relationship is the same and your fixed relationship is the same, no matter how far you stretch this left and right, you will always get four numbers of module. So this is sort of they have a mechanism for that in the paper if you're further interested. And this is a one-dimensional example, but this works for two-dimensional examples as well. On the bottom, have a two-dimensional example of you have a two-dimensional analog of this smoothly varying relationship and a non-varying relationship. And if you overlay them together, you have these kind of grid-like modules that arise. So that was the Konad Chandra Feat 2025 paper.
I mean, that's pretty interesting. It's not obvious to me why combining those two signals would result in this sort of sudden transition between one module and the next. I'm sure it does. I'm just looking at it, going, "Hey, why would it have those sudden transitions?" You know, I have a continuous variable and a static variable, and why would it do this? I'm sure it does. I just
Yeah, yeah. So this is the part where I go, yeah, this looks interesting. And if
okay.
we want to do a deep dive,
No,
It's
that's right. It's
I think it's basically they have another figure in the paper where they've visualized basically the green and the orange curve over time layered on top of each other. And so basically, since one varies smoothly along the scale and the other one locally, the it will get pushed into different local maxima or minima, depending on how you look at it, at different points in time. I can
no. Now
um,
you know what? Now I can imagine. I can imagine a
like they're
mechanism
bas
to do it.
yeah, they're
Yeah,
basically
it's
different basins, and then
right, and
um,
it's gonna try to fit everything into one basin until it can't, and then it goes into the next one or something like that.
yeah,
Yeah, I
yeah,
got it. I got
kind
it.
of.
I got it. You don't have to go any further. All right. Well, this is interesting.
it's a thing.
Interesting. It's interesting. I mean, I'm almost to make of it yet, but it's interesting.
So here's some of the just predictions that come out of this mechanism as precise period ratio values, topographic grid patterning within modules. Uh, you get these smooth spatial gradients, but no discrete steps. Coexistence of fixed and graded properties. Let me see if there's anything worth highlighting. Self-scaling with system size within and across species. That's the big one. Grid boundaries are orthogonal to module axis. I think it just matters like the dimension where your smooth thing is varying, because this can be like, like gene
So,
expression or chemical density or
so
something.
was this a purely theoretical paper, or did they see some of these things already? Did they say, "Hey, we saw these two kind of functions, and we tried to figure what they did," or did they just come up with something that says, "Hey, well, we hear the mechanism. It could explain this. We should look for these things."
Um, if I recall correctly, it is a kind of computational model first of how could this emerge, and it was. If I recall from the presentation, they were like, okay, we're gonna try this smoothly varying thing, and we're not getting modules. Let's add this uniform thing. Oh, and
Okay,
now we have modules. So it was more of that type of exploration, uh, which is
so
computational first.
it wasn't empirical results yet. They're just saying, "Here's how it could happen."
Right.
Yeah, I think, yeah, they say in the abstract that they make testable predictions, but it's not
Hmm.
like they modeled the data itself.
Well, sometimes they could say, "Oh, these cells, we've seen cells like these gradient cells, or we've seen cells like the Mexican hat cells, and something like that." But that's okay. Got it. So somebody came up with a mechanism, says, "How could this come about?" And they propose this mechanism, and they say, "Here's how we can test it."
And and then the one last thing I guess I'll highlight is this mechanism second from the bottom, simultaneous, not sequential reemergence activity in all modules after suppression. So like because this is all does all sinusoidal wave activations, when they suppress it, like when they after suppression, the entire grid structure reemerges right away. There's not like a progression or takes a long time. It's like everything just jumps back into a grid immediately.
Oh,
Uh,
this is interesting. So this is a very interesting some question that just pops in my head. I was under maybe the incorrect assumption that this is a mechanism that could explain how these grid modules came about, but I assumed it more like a development issue, like, oh, during the development of the animal, this mechanism would explain the emergence of these grid cell modules. But what you just described now, Tristan, suggests that this is a dynamic thing. It's happening all the time. That the grid cell modules aren't like fixed. They're relying on this mechanism all the time to do this. So you can disrupt the grid cell modules if you remove these signals.
yes.
Oh, it's interesting. Hmm.
So
Oh, and I think one thing that they like the bridge between data and their model, one thing they noted was that the scale changes between different modules matched what was found in biology
Mhm.
before, like
Mhm.
how much the scale changes between two neighboring
Yeah.
modules.
Would it be possible using this mechanism to come up with, like, instead of 8 grid cell modules, 20 grid cell modules? Is it, I mean, by changing the Mexican hat function in some way?
that's my understanding. It's the how many modules you get is controlled by like those two parameters or something like
Well,
that.
the continuous gradient, the only thing that it can control is the scale of the maximum value. Um, or the, but the other one seems like the Mexican hat one would be the more critical one.
Yeah, I
Um,
think the one that controls number of grid cell modules is basically the width of the one that stays constant no matter.
right, the Mexican hat one. That's what I was thinking.
Yeah.
Right. That makes sense. That would be the one that does it. I'm just wondering if that mechanism could, like, oh, we could produce 100, or we could produce two, or is it sort of like the sweet spot of like, yeah, 8 or 10 is what you typically get, or something like that. If that's not important, I'm just curious. You just wonder, like, oh, here's an interesting mechanism that neurons are doing that I've never heard of before, or at least never heard proposed before, and thinking, well, what else could it be used for? I don't know.
Yeah. And so afterwards I specifically asked like, okay, this is pretty scale free. Like, is there anything you can think of that would prevent this from happening at the scale of a single cortical column? And like, no, it's a generic mechanism. So
Right. Well, that's another really interesting point. We've always assumed that this at a cortical column there'd be one grid cell scale. That's what we kind of always thought in the past. And then the changes in the size of the receptive fields of the different cortical regions, we speculate, hey, maybe that's like the different grid cell module scales. Like, you go from V1 to V2, you get this larger spatial scale. If this mechanism were true, that would seem less likely, because then the gradient would have to these two variables would have to expand over the entire neocortex or regions of the neocortex. So it would be more likely you might get different scales of grid cells within a cortical column or cortical region itself.
Which would
I
be
don't think
more useful to, like, if we only
right,
have one
probably, yeah,
module, then
right. Although it does strike. I mean, it doesn't seem like there's enough neural tissue in a cortical column to get multiple grid cell module scales side by side. At least not many of them.
yeah, I think the main issue is that the scale at some point it breaks down because you don't have enough neural resources anymore
Right.
to keep the local dynamics going. So I think, I didn't read that part too carefully, but I think they tested at what point it breaks down. I mean, it's all simulations, so it's hard to translate
Yeah.
to neurons and size of cortical columns or anything. But yeah, I think that was basically the breaking point. But you could imagine maybe that a couple of grid cell modules would fit within a column, and given that less neurons are dedicated to each module, that makes them maybe a bit more noisy in the path integration, and there's less redundancy in the system, maybe.
Yeah.
But
The other weird
maybe that's
thing,
enough.
I don't know. The other weird thing is, papers we read a couple weeks ago, we were shown that the grid cell modules overlap, right? They weren't intermingled. They were sort of overlap stacking. So that's what the paper said. And so if the different, this is in the entorhinal cortex. And so in the entorhinal cortex, if the grid cell modules are overlapping, sort of like a bunch of dominoes that fell on top of one another, so they're kind of overlapping but moving along in some direction, that would be interesting to say, well, where is this, where would the linear scale variable, it can't just be stretched along this tissue. It's like, because the modules are overlapping, it wouldn't be like the picture you just showed, Tristan. They would be kind of, yeah. You follow up. It's an interesting question. It's
Yeah.
it's like those different modules would be overlapping on top of one another. And then it questions like, well, how do these two variables work in that case? It's like strange. It could. It's just weird. Okay. Oh, that's interesting.
So that's the published paper that covers that particular mechanism that Vivian referenced, that we've been
Was this in
reviewed.
a poster or was this a talk? Or how
This
did
was
you
a
come?
this was one of the main talks.
One of the main talks.
Yep.
So it got high profile.
Yes.
Okay.
Okay. Next topic is what's necessary for place cell remapping. Reason I thought that this might be interesting to highlight is having to do with, I'm always thinking about how do we switch objects? How do the cortical column knows to switch reference frame, that sort of thing. So this was kind of tangential related to, here's a mechanism for how what's necessary for place cell remapping to happen. So what I mean by that is these folks they picked out two circuits out of the vast complex circuit that goes from sensory input to the hippocampus entorhinal cortex. These two circuits they're focusing on the left are just going from sensory input to LGN V1 lateral visual cortex. This is mice. And hippocampus entorhinal cortex, and the other one is the superior colliculus pulvinar lateral visual cortex. And so those are the two. They did some analysis and they focused on these two. And so the experiment was as I believe it was like a mice running along the track, and what I want you to get out of this, they were recording. These are 140 cells recording, and they refer to these as place cells in CA1. And the way they are demonstrating the correlation on the pictures here means like when they sorted it out, when it's sorted, it means that they have like a coherent mapping. And so what this means is when the mouse is running through, and it's in a lit environment. Uh, there is a consistent mapping between when it goes to light A and light B conditions. So those are the same mappings of the place cells. That's how this should be read. And then dark is incoherent, but if you sort the dark ones, you'll see that when it's actually running the same maze in the dark, it uses a different mapping. And so the high level point here
What's the difference? What's the difference between light A and light B? Or is
I
that a
don't
time difference? Is like they do light pass and then a dark pass and a light pass and a dark pass, or is it something else?
I don't remember. I don't remember, honestly.
Well, it could be the experiment is they do the experiment in the light, then they do the experiment in the dark, then do it in the light, and then they do it in the dark again, which could be
That
A,
sounds
B.
familiar.
Okay, or it could be like they have like light A is, I don't know, disco ball. I don't know what light B is, something else.
Oh, no, no, it
Oh,
was like A and B were like trials, but
okay, trials.
everything was
Great,
like kind
great.
of like the same. Everything was
Got it.
kind of the
Right,
same
right.
track.
So they're just going back and forth between the two trial conditions.
Yes.
Got it. Interesting.
Um, and so, and I don't know, it says 20 minutes, so I don't remember how long it's exploring or running around and whatnot. The idea is that when it's going through the light A condition, there is a place cell map activated. And then when it goes to dark A for 20 minutes, it's not, at least this particular map is not activated. And then when it returns to the light condition, the activity is also is correlated to what it was experienced, what was recorded in the light A condition. So that's sort of how that's read. So at the high
I'm
level,
guessing they're saying they're using the A sort on the B data. Because that's the only way that would
yeah,
mean that, right?
Yeah,
yes,
yeah.
yes, A sort
Like the
on the B
same
data. Yes.
sorting.
Yeah.
So you see the same thing emerge, which means similar activity should be happening.
Mm.
Oh, well,
Uh,
what surprising to me is that there's a different place cell mapping in the dark. I mean, I would have expected, oh, maybe there's no mapping in the dark. The animal doesn't really know where it is. Or you would say it's the same as in the light condition. But the fact that there's a completely different mapping in the dark strikes me as odd. Um, that's like saying, when I've talked about, I walked from my bed to the bathroom in the middle of the night. Well, if I do it in the light and then I can do it in the dark, um, and it's saying, oh, I have different place cells when I'm in the dark than in the light. That's weird.
Yep. Like,
It's also really surprisingly stable for like real mouse data. That B
Now that I
sort.
think about it, it's maybe not so weird. Um, Place cells aren't good cells. They're to sort of they're environmentally clued. So when I walked between my bed and the bathroom at night, I I'm looking for different clues to know where I am. I'm touching walls and corners, um, things I wouldn't necessarily look for if I was walking in the light. You know what I'm saying? It's like the things that matter to me that to navigate in the dark are not the same things that matter to me when I'm navigating in the light. Even though I would have the same location in my grid cell reference frame, I could have different um, place. These would be physical, tactile place cells in my brain versus visual place cells. What I'm saying? Ones are driven by vision versus ones are driven by touch. Um, okay, it's interesting. Sorry, I'm thinking out loud.
yep.
But
So, so the point of this is to establish that there is, when this slide was presented, was to establish that there are different mappings in the light and dark conditions for the follow-on experiments. So the idea was to figure out of these circuits again on the left, what do you actually need to retain that mapping? So again, going back here, this is a healthy mouse. And so over here, they lesion the lateral, what is it? Visual cortex, and place cell mapping remains. So that's the highlight here. So you can lesion lateral visual cortex and place cell mapping remains. You can lesion the V1, and I'm sorry, let me rephrase it. I said it exactly backwards. You can lesion the lateral visual cortex and remapping disappears. Remember, dark A and dark B were supposed to be different. but you get the same activity in light, and dark. It kind of looks like the same activity, so there is no remapping occurring when the lateral visual cortex is lesioned. Excuse me. So this would hint that the lateral visual cortex is necessary for remapping of place cells to occur in this experiment.
Makes sense.
Okay, sorry for just saying it exactly wrong way at the beginning.
That makes sense, Scott. I don't know what lateral visual cortex is versus V1, but
I
I don't know.
I definitely don't. The other
Yeah.
part was interesting is the circuit is when they lesioned the V1, the remapping was retained. That is, you get different activities when you were in the light versus in the dark, or the light and the dark. So the conclusion here being that V1 is not necessary for remapping of place cells to occur. And then kind of narrowing it down, they lesioned, although like in the previous experiments, they literally cut out the pieces of brain tissue. Here it was lesioned in a using a different mechanism, but they lesioned what are called white field cells between the superior colliculus and the pulvinar in this part of the circuit. And that was sufficient to destroy the remapping. And so ultimately, this is the narrowing it down. The detail is no light or dark remapping without white field cells. So I don't know how relevant this is, etc. It just, again, going back from my outsider's view was we care about remapping things, and
Yeah.
that can be taken away if you lesion white field cells between superior colliculus and pulvinar.
One thing that jumps right out at me is if the animal's in the dark, how does it know where it is? And it's got if it's completely dark and there are no other clues, it would use its whiskers to know where it is. Um, I don't know if they address this issue in the paper. I mean, you can do path integration, but that fails quickly. So for the animal to know where it is, for these place cells to become active without any light, the animal has to have some input, and so that would be presumably whiskers touching walls and things like that. In which case, I don't think the whiskers are represented in the circuit on the left of this diagram at all. So if you go back to this one here, where the remapping occurs, uh, they don't show the second row of those maps, which was what was the second row? It's showing that the dark A and dark B had their own sort, right? Yeah, that one.
This one.
Yeah. But if we assume that, you know, again, it's to the comment I made earlier, the dark A and dark B mapping might be somatosensory, might be whiskers. And so anything you do to the visual cortex may not affect those whiskers. So all I'm saying is there could be a lot of confounding variables here, and I don't know if they address them in their discussion section or talked about these things or not. Because just ablating or disabling one visual region doesn't mean that rat has no idea where it is. Anyway, these are questions. These are not criticisms of the paper, and they might be addressed in the paper. But just from looking at this here, it's like, oh, well, what's going on? How does the rat know where it is? And are you accounting for the fact that it has other sensory organs? It tells it where it is.
Okay.
Interesting. Well, I think it's fascinating to me that there's a remapping at all between the dark and the light. So that's new to me.
Nice. Okay, next. Superior colliculus has cylindrical modules that define an egocentric action-spatial map. I'm sure this is going to be a small topic. So This was a new to me thing. So like literally this is that sentence is the topic highlight that I wanted to share. They didn't, here are the three papers. There's a paper 2018, 2019, 2024 down here in the slides, where they talk about it, but it sounds like it's a known thing. I just don't know if it's known to us. So just wanted to highlight
Well,
that.
I think what we have talked about, Tristan, and certainly on my case, without any expertise, we've talked about there is a known egocentric map across the surface of the superior colliculus. So you touch on some point in the superior colliculus. It represents a point in the space of the animal's head. Some place in egocentric space. So I think that's been known, at least at a very top level. And personally, I don't know any of the details of that. But this idea of cylindrical modules is very interesting. So I don't know if you're going to go into that at all.
Nope, not at all.
What's an action spatial map is versus just a spatial map. I do think it's like if you touch the part of some point on the superior, it's like an attentional mechanism. The animal would attend or turn its head, I think, to that point in space, some point in space relative to the head. I think that seems familiar to me. So maybe it's referring to that.
Yep. So
Um,
so this is a
Is,
preamble to the talk, and this is a preamble to the next topic. Um,
is this supposed to be analogous to cortical polyps? Or okay, because they say cylindrical models, it's like so like a
right. I
cauldron.
don't. Yeah. Well,
there's definitely talk of modular structures.
Okay,
so
okay.
so that modular structure could be something visual, like that image in the black image in the upper left one of the slides suggests that they're seeing these modules as a physical thing,
Okay.
right? You can see that there. It looks like they're highlighting here. They're doing some kind of staining or some sort of imaging thing, and saying, look, we can identify these. That's one way you can have a column. The other way you can have a column is you try to do what Mountcastle did, which is, say, oh, they physiologically they behave
Like
differently, and you're some
functional.
sort of transition, right?
Yeah.
I don't know if they have that one, but the image there showing the stain or whatever that thing is suggests that they're at least identifying it physically. Also, the next picture over says the column to column spacing is 130 microns, which is pretty small. That's not cortical column size. So I don't, it might be columnar, but it's not like a cortical column. At least it's unlikely because it's so small.
Yeah.
But interesting question. What's the difference between these columns?
So that's all I had to say about that. But this was a preamble to the rest of the presentation, and the rest of the presentation was spatial salience as motor readiness. And they called it a novel premotor framework for spatial salience. Uh, we've talked, I spent a lot of time on salience, learning salience, working with Scott. We're talking a lot about attention. This is why this seemed interesting. And we talk about a lot in our context is sensorimotor. There's a lot of reasons why this was interesting. So the main thing that they're highlighting here is the two schools of thought about salience, and they're not disproving any. They're just highlight the intent is to highlight the what it sounded to me like a um, less prolific school of thought. So they acknowledge salience as a sensory modulation, where the salient things are getting magnified by the sensors. But what they're going over in the presentation here is salience as spatial motor readiness. So instead of having sensory magnification and focus, is you're ready to do an action, and salience is just queuing up that action for you to do and making it easier for you to take an action. This is
Both,
the
both of those could be true, right?
Yes, yes, yes. They
right.
said it's not one or the other. It's just they're just talking about the second one up here about motor readiness.
Right.
And
And
this
to
is
me,
the
that one would be most likely associated with the colliculus because it's like that attention mechanism I just mentioned. Like, like you see something moving off to the side and something salient over there, you're more like you're gonna, your body's gonna turn and look at it. Where that doesn't seem to be always the case in the cortex. So,
And
okay.
so and so um, so let me see if I can walk through this slide in a coherent manner. So what you see on the right here, what they're talking about now is this novel premotor framework mechanism that they're describing here. This is a mechanism that happens inside those columns that they've talked about before. So the columns are taken as for granted, and these columns apparently have 3 layers of cells: the CCK cells, SST cells, PITX2 cells. I don't know what they do. I don't know what these things stand for. What the CCK, well, I know what they do because they talked about it, but I don't know any more detail aside from these acronyms. The CCK, when you have what they're talking about here is the ethological saliency. So you're getting some sensory input from the outside, like this was a mouse hunting a cricket, and that was the analogy they were making. And so as the cricket is moving, there's a video here, as this cricket is moving around, this activation in the CCK is essentially tracking the cricket throughout the field as well. So if the cricket is on the left, the field is essentially highlighting where in the sensory field the cricket is. So you have kind of this like your first level of saliency. Then this SST thing, all the activations are the bluish things, and that suppresses saliency. So it's like a SST layer is like a saliency suppressor. And what happens is the CCK activations suppress this. So let me use better vocabulary. Excuse me. Excitation and inhibition. The SST layer is inhibitory and inhibits all saliency. CCK is excitatory and it is excited wherever there's an ethologically salient input into the field. And this CCK excitatory. Nope. Okay. I keep on resetting because I keep on saying the wrong things. So please forgive me. So once again, to reset everything in this network and this 3-stack is inhibitory. So sorry, I got confused by the excitatory connections. Excitatory is reserved for actual actions later. This novel mechanism is all inhibitory. So let me start over. So you have the CCK layer where inhibitory neurons are being activated, tracking salient, ethologically salient input to the world. So wherever the cricket is moving, there's this field of inhibitory neurons that is activated. The CCK inhibitory neurons then inhibit all of the SST inhibitory neurons. So it essentially carves a hole through the inhibitory SST layer. So SST layer inhibits, but the CCK neurons inhibit the inhibitors, therefore punching a hole through the layer. And because CCK inhibited SST, then these PITX2 cells now, wherever the saliency hole exists, now these are primed. They do not cause any actions, but they allow for action to happen easier. And this goes back to they allow, what is the neuroscience language where it's easier for neurons to trigger. So
depolarize.
Thank you. So they depolarize them so when an action potential, excitatory potential comes through, they depolarize faster in that particular area that those neurons map to. So back to the column diagram over here. Again, everything, this entire mechanism is inhibitory and no action actually take place unless you get feed-forward excitation coming in through the column and activate this PITX2 neurons, which have then been depolarized to, sorry, what was the term?
Yeah,
depolarized.
Yeah, depolarized, so that these are the ones that actually are more likely to trigger the action input as opposed to all the other ones that are being inhibited. So sorry I stumbled through it, but that's sort of the mechanism that they're talking about here.
Can you leave that picture up there? So it says at the upper right, it says spatial coherent column spelled without an N. Interesting. Approximately 20 degrees. And then they show like a gradient moving in some direction. Well, this may be too much to ask you, Tristan, but when they say column without an N, approximately 20 degrees, is that representing how much space this column represents, or the offset of? Do you have any idea what that means?
They didn't highlight the 20-degree aspect in the talk.
Okay.
I didn't hear them
And then
bring that up.
spatial coherency. Well, there's a lot of mystery. There's a lot of like, oh, look at that picture. It suggests a bunch of stuff. I have no idea what it's actually doing. Okay.
Well,
Well, this gets back
because
to the question, what's the column, right? How do
and
they define it?
and I can't tell if the 20 degrees is like within this column or whether that's some sort of sensory 20 degrees visually.
I assume it would be somehow sensory 20 degrees, but I mean, we someone asked earlier, like, oh, what does this column represent? Or what is this column? Well, these are clues. Those little words are clues, and those pictures are clues.
Right.
But we don't have to worry about it. We don't know right now.
Yeah. Yeah.
Um,
And basically, if you try to imagine a video on the left playing, the cricket is moving around and all the holes are just tracking each other. So, right.
yeah.
So that sort of was what the demo was. And it's all inhibitory, but I thought it was interesting that inhibitors inhibit inhibitors so that activation can happen. Of course,
This kind
nature
of
found a
this
convoluted way of doing
well,
something.
this kind of language appears very, very often in neuroscience, and certainly there's some truth to it. But I believe it's often, not necessarily this case, but often it's quite misleading because there's lots of neurons here. They're very complicated. Most of them have, thousands of synapses or many synapses, and they're almost certainly doing something more sophisticated than they suggest. Um, but they can say, oh, but these are inhibitory cells and these are excitatory connections. Therefore, the excitatory connections activate the inhibitory cells, so they're inhibiting something. It can be a very, very again, I'm not accusing this particular paper and these authors of this, but often these kind of language of disinhibition, inhibition, and these things are often the language used because they don't have any other way of describing things. But typically, it's a very, very gross description of what's going on. Usually, there's something much more complicated happening on top of it, so we can't worry about it too much. And this kind of multi-layer disinhibits this, which disinhibits that, and so on. You see that everywhere. It's all over the place. There's in cortical columns. It's everywhere. So we shouldn't worry about it too much. It's interesting on the picture on the right, they show these little columns having both like an inhibitory side, which is colored, and then the feed forward excitation side parallel to it, suggesting these cells are the way the picture is drawn suggests that they're sort of segregated left and right in that picture. I don't know if that's true either, or are these cells intermixed with each other?
Yeah, I
Which
don't know if they didn't say whether that's like
right,
anatomical distinction or whether they're
right.
just diagrammical
Right. Well,
distinctions.
this is one of those situations you'd have to go back and read the original source papers and go into their methods very carefully. It's a lot of work to figure that out. But there's so many things in neuroscience where you can't get everything you want to get out of a paper. You just have to like, oh my God, there's so many questions it leaves open.
And so this reminded me, so this was sort of also felt a lot like a covert attention mechanism.
Yeah. Well,
Um,
they showed that in the earlier slide, didn't they? Um, they're seeing the
yeah,
upper left. They say overt movement
covert
and
spatial
covert, right?
salience. Yeah.
As you see, right, right.
So,
Okay.
um, and so again, they're not saying salience as sensory modulation isn't a thing. What they're adding is a specific mechanism for salience as a spatial motor readiness.
Hmm.
And Daniel de Malmazet was the main investigator of this. So these are the people, the lab members, to reach out to farther if this is of interest. Okay. Uh, Next thing, this was a comment made by a speaker as a side comment. Then I asked about like, what do you mean by place? And it's like, there's place cells everywhere in the neocortex. And I was like, asked him afterwards, what do you mean by there's place cells everywhere in the neocortex? And so there was what am I doing? Okay, yeah, I'm quoting a paper. So there is a paper, and I'm just highlighting it here. I'm not going over what it found, but uh, we can investigate it farther if needed, but this is the quote. Within our brain-wide neuropixels recording approach, we find that many regions outside the hippocampus, including striatal and thalamic regions, also encode spatial position.
This quote's from a paper or from
This one, it's from this. Spatial navigation engages a distributed neural representation. So
Mhm.
that's all I have. I was just more like I heard a trigger phrase of place cells are everywhere in the brain, and so would be the farther thing to look at. But
Well, we had those papers that we knew from the lab in China. I can't remember the name of the researchers who found place cells and grid cells and head direction cells in V1 and in S1. And this, of course, this is supported by our theory, right? Our theory says that there should be place cells not just representing places, but also locations on objects, which would be like, so you can argue that um, border ownership cells are a type of place cell. That's what we would say. So it's interesting that this is maybe becoming a more widespread view. Well, they haven't gotten far enough yet to say, oh, it's more than just places and environments. It's also places on things, which is what they'll find, I think, if they keep looking at this stuff.
Okay.
So it's good. It's good news that people are saying these things.
So I've got two more to go. Dynamics of remapping across the spatial navigation circuit. What does that mean? So this was a poster from Modular Lab. It was Joe Carpenter, and essentially, I don't have any pictures because I said please no pictures. But this is the summary from on the web from the poster session. Like spatial navigation relies on the coordination between metric representations from grid cell modules, spatial anchoring by place and border cells, and directional reference frames encoded by head direction cells. Reason I'm highlighting those three things is because this poster was about investigating in rats whether uh, whether those are the things you need in order to get coordinated remapping across different places in the environment. Uh, that's gonna say nothing. And so what I'm gonna go over next is go into and so this is the investigate.
Justin, can you just go back a slide? I want to finish the paragraph. I'm just trying to read
Sorry.
it. Do you mind? I'm doing active high-speed exploration. Okay, so it's talking about remapping and the dynamics of remapping. Okay, thank you.
And highlights that all these three things are tightly coupled for the remapping to happen. And so this is actually experiments in live rats recording these things and demonstrating that there is a very tight coupling between these three things in order to make the remapping across the spatial navigation circuit happen.
Of course, this is these are, if it's not obvious to everyone, these are directly analogous to what we do in Monty, right? Our head direction cells are the orientations we talk about, orientation cells. Of course, we have locations, which in the brain are done by grid cell modules. And then place and border cells would be sensory derived input. That would be to like our layer 4 in some sense. But based on what you observe, you recognize features in the environment. Those are associated with places, and then in layer 4, and then we have our head direction cells in layer 6B, and we have grid cell modules in layer 6A. So just make sure everyone catches that, just in case we missed that.
And so I can't I can't share much more about this here now, but this is the animal experiments demonstrating a computational mechanism, which I'll talk about next. And so Jo Carpenter is a PhD candidate in Moser Lab. So she's the one investigating this if we want to reach out for further information. But it's investigating essentially this vector hash mechanism of FeedLab that is a computational model for how these three things are coupled together in order to make remapping across spatial navigational circuits happen. And so since I don't have these results, I can go over this vector hash thing of which Joe is. I've asked, so did you demonstrate that vector hash work is actually exists in animals? And the answer was like pointing at the poster. So that's the interpretation. This is the vector hash mechanism. This also, I didn't write this down here. Sorry. This comes from FeetLab again. So the original, the very first topic I talked about, Ila Feet from MIT, talking about emergence of grid modules. This is the same lab, and same Chandra was the lead author on this. So this is again the computational model, computational model. Let me kind of go over what this is kind of the underlying computational model that's investigated, and I'll show different varieties of. So what vector hash is. Over here in this is torus, right? What's the plural of torus? Tori.
Yes.
So these tori represent different grid cell modules. So in this picture, we have like 3 grid cell modules. And so in all the schematics, you'll see there'll be, this means grid cell modules. And these self-pointing arrows just kind of point that the grid cell modules interact with themselves. Then what we have in the, whatever this color is, these are the hippocampus. This is the essentially place cells. So it's called hippocampus. It's referred to as H everywhere, but the idea is these are place cell activations. And so you'll see there's a highlight here of a scaffold, and essentially there's a computational scaffold between all the grid cells and all the place cells. And the important part is that this is a stable scaffold, i.e. the connections here are pretty much stable and the same. It's more, the reason it's called a scaffold is because it doesn't change that much. It just, once established, there are these dynamics that I'll talk about between grid cells and place cells in this computational model that essentially stabilize each other. So what ends up happening on this scaffold in this computational model is if you give it a place cell input, it will find the corresponding grid cell input and vice versa. So I'll talk about that more next. But I just want to highlight these things are grid cell modules, 1, 2, 3. These are meant to be place cells. There's this V thing component that you'll see, and that's sort of like your directionality signal that this is in the computational model. This is however you're moving through space, this will essentially be driving your grid cells to change because of your movement in a space sort of thing. Now, it might get confusing because again, I've mentioned place cells stabilize grid cells, which stabilize place cells. So there's a lot of recurrence here. V specifically drives it at the grid cell level. And then lastly, there's this non-grid cell cells. So there'll be this green sensory layer. And these gray connections here are learned connections. So again, the dynamics, so you see this heteroassociation. So you can think about the scaffold consists of 2 units, the grid cells and the place cells that have internal stable dynamics and they stabilize each other. And I'll talk about more what that means, where the grid cells can be driven by a direction signal. And then separately, there's this heteroassociation thing between sensory input and just think of it as the scaffold. Just think of it as 1 unit. And so although it's directly connected only to place cells. And so what you get is, and this is learned associations. So if you can think about it, scaffold is sort of like, this is how you get your metric space. Here's your sensory input. And these learned connections are, this sensory input belongs in this sensor, in this metric space, location at the metric space. Think of it, that's what heteroassociation kind of loosely means here. And so, and you can see right here, plastic Hebb-like learning, fixed non-random, and fixed random. So again, I want to highlight the connectivity between grid cells and place cells in this computational model. These are, there's fixed connections that are non-random and fixed random connections. And so
what does that mean? What's the difference between?
What it means is, as you're using it, the weights don't change.
Yeah, but that would be fixed for both of them. So what's the difference in the random and the non-random?
I honestly don't remember. I'm sorry.
Yeah, no problem.
The important highlight that I remember is the fixed version of it, where they don't
Yeah.
change. I I remember when
Maybe it was like the connectivity. Some are connected by randomly and some are deterministic for whatever reason.
yeah.
Well, but if it's fixed, then it's
Uh,
how would you tell the difference between them? I don't know.
Maybe you do one randomly at first, but then to get the backward projection, you have to infer that based on the random
But
forward
then,
projection, so
but
it
then it would
matches
be.
back.
I I don't like because I've generated these scaffolds computationally before, and it was essentially all I was doing was everything was random, and they still worked. So as long as they were fixed and random, as long as they still worked. The one caveat I would say is maybe you need. So what will happen is if all of these are random, what you will get is that. Uh, so let me. So these grid cell modules can represent a final number of locations, right? Like there's because these are all discrete, so they're gonna represent a final number of locations. If you have both
Final, you mean finite?
finite,
Finite
sorry,
number?
excuse me,
Sorry,
finite,
finite. Yeah.
finite. Before you go back to the same place on the torus, even across all the grid cell modules. Uh, so where the distinction between random and non-random might be relevant is when I was generating these with all the connections being random, I could come up with connectivity where not every location would be used. Like there was not a location to every possible grid module location. And so maybe this is, I'll have to go back and reread the paper. It's been a minute, but maybe this has to do with making sure that um, there is a mapping to every grid cell because essentially the number of locations in this grid cell modules is the limiting factor of the entire system here. And so maybe this is a technique for making sure that all of the locations are used in computation here. What I've done in practice is you can make all of these random and just test and ensure that there is a corresponding mapping to every grid cell. There's none repeat. And so you can empirically just sort of determine this connectivity. But what's nice about it is you can spend a lot of compute finding a scaffold, and then you have a fixed network, neural net, weighted network that you never have to change or learn, and then will give you the scaffolding dynamics that will do some pretty cool things on next slides.
Okay.
But
So there's a lot to talk about in this slide, but
yeah.
I'll wait till you give you the rest of it and we can come back.
Okay. Yeah. And then we can come back to this.
Right.
Okay. So first use of it, and notice some pieces are missing. We're not going to have this V thing here. And so this is the grid cells that I talked about. This is the place cell. Like I said, it's going to be labeled hippocampus everywhere. And this is the sensory thing. So again, grid, place cells, sensory input, grid, place cells, sensory input. So hopefully that continuity retains. So here's how they got, this is what they've done computationally with item memory. And this is the interesting part about this. This talks about the scaffold here. So notice in figure 8, we don't have actually the sensory part. This is the part of I was trying to say before, so I'll repeat it again about there's a dynamic stabilization between the grid cell layer and the place cell layer. So what will happen in figure B over here, you can have an original grid state. So if you start your dynamics from here's the state of grid module activations, then that will activate place cell module activations in some way. Like they just correspond to each other. Again, it's a scaffold. These are fixed relationships. If you add noise to the hippocampal state, to the place cell state, you add noise, you're now gonna get back a noisy grid cell state. And these self-referential loops here, where the grid cell is kind of talking to itself, there are internal dynamics that essentially takes the noisy grid cell and then stabilizes itself again with the scaffolding into a non-noisy grid cell.
These are simulations, right? These are all simulations.
This is all computational stuff. Yeah.
Right.
Yeah. Everything here, vector hash is all computational model simulations, Python, matrix
I mean,
multiplications.
on the surface, this is not surprising at all because there's this the assumption on grid cells is that there's these inhibitory surrounds. The system wants to lock into, a set of bumps across the surface. It's the not looking regular. Um, it's not a normal state for the grid cell. So it's just the way the neural mechanism is. It's just going to want to settle in on a set of bumps. And if you disturb it, it's going to go back and settle on the same or a different set of bumps. Um, I mean, it's not a surprising result, that part.
Yeah.
It's like,
And so the main thing here to take away, I think, is the dynamics between the place cells and the grid cells stabilize it. So if you give it a noisy place cell, the dynamics will take you to noisy grid cells, and then which will then activate place cells and grid cells, where you're going to end up the non-noisy state.
unless your noise was sufficiently large, then it can lock into another state, right?
And then you'll recover a different state. Yeah, exactly.
Right. This is the sort of saying I walk into a room, I'm not sure what room I'm in, there's some conflicting data, then I say, oh, I'm in this room, and you lock onto something, right? That's all it's doing.
So
That's
this
what
is sort of
saying.
the essence of this scaffold. It's the point is it's a stable scaffold. You can add noise to it, and it will clean up the noise out of itself. That's the main purpose of it. So now talk about spatial. Okay, so why did I say this item memory? So let me just talk about the upper right corner over there. If you add a sensory layer in there, and I mentioned, let me just show here. These connections are in light gray, which means they are learned connections. And so what ends up happening is when you get sensory inputs, they will trigger some sort of place cell activations, which will then do the stabilizing dynamics we just talked about. It's going to activate some noisy grid cell activation, which will then clean up the place cell activation, which will then make a prediction for what sensory input you expected to see. So like there's this dynamics here where you get noisy sensory input, you hit the scaffold with it, and the scaffold tells you back, this is the clean sensory input sort of dynamic. Very much this.
The animals recognize the environment, and once it recognizes the environment, it can predict what it's going to see or feel or things like that. Right.
All right.
So
that's what they're describing here. So for spatial memory, we're adding this vector component where we're going to be driving movement, so it's not just place cells activating the grid cells, but also you're moving through the environment, so your grid cells are going to be changing the location, and so there's a driving factor from there. And so again, the highlight, there's a scaffold and there's this heteroassociation sensory that I just hinted about previously. And this is the figure B is the cool figure. And so what you get is, this is once you've learned. So once you've learned an association. So here all the way on the right is the memorized pattern. So you had some sensory input that you threw in the scaffold, and then the scaffold learned to output back the same sensory pattern. And again, one thing I want to highlight is these are used as images because they really work well for communicating the idea, right? But these are just bits, patterns, SDRs, whatever you want them to be. It's not saying we see with our grid cell modules. It just means this pattern, it's easier for us to process the pattern is recovered or not. So I just want to highlight that. So you get a noisy pattern where you like obscure the center of the bird, for example, or something like this. This gives this sensory, like on the figure here, number one, it will activate the place cells in some sort of noisy pattern. Then two, the place cells will activate some sort of noisy grid. You get grid self-stabilizing dynamic, which will clean up the grid, which then the cleaned up grid will activate cleaned up place cells, and then the cleaned up place cells would then activate whatever the learned recovered sensory pattern is. And so you can memorize a pattern, then you can give it noisy input like this, and this scaffold will recover the pattern through this dynamic heteroassociation. So again, the scaffold is just all the weights are fixed. It's literally just a network that cleans up noise at a location. And the dynamics, all the learning is happening between the scaffold and this sensory layer.
You could argue the opposite. Well, not opposite, but something different. You could argue that the grid cells aren't there to clean up the place cell representations. The grid cells there are specifically for path integration. I mean, you could build an associative memory between the hippocampus and the summary, between the purple and the green boxes here, that had all the properties you wanted. But the grid cells add this extra component. It allows you to take a movement vector and say, I should predict what I'm going to sense at some new location, even though I haven't sensed it yet. So
Yep,
it's
that will happen
not
too.
denying what you said, Tristan, but I think nature of, it's good. It's a good reason to say that grid cells evolved not for cleaning up, but for prediction after movement, which is a form of cleaning up. It says, oh, anyway, I just don't want anyone to think like, oh, grid cells are just there for cleaning up. No.
No, so yeah, that's a few slides ahead. So
Okay, I'm sorry.
Because that's exactly what's going to happen is they're going to drive a vector to move grid cells somewhere else, which will
Right.
then generate a new clean place cell pattern, which will then make a sensory prediction of what you should be seeing.
Right.
So this is the details of this is kind of gives you more of a mathematically spatial intuition about how the cleanup works. But in essence, you get a Q pattern. The grid cells essentially divide all the noisy patterns, and they essentially just basically they have sinks, right? So as long as you're close enough to a specific noisy grid cell pattern, the dynamics will put you back in the center of it. So when your predictions are always clean. The relevant part here is if you make a movement, you are always making like these clean. All your predictions are the clean version. You don't make noisy, crappy predictions, I guess is the highlight, because they're generated from the kind of the ideal grid cell state that's stabilized. So all the noise that comes in either from movement or from sensory perception gets cleaned up by the grid cells because they have these location sinks essentially. It's like no matter where you're gonna end up noisily, you're gonna only activate one grid kind of pattern. And because those patterns are discrete, then that discrete pattern will then lead to the cleaner prediction of what you should be seeing. So that's sort of the approximate but identical sensory reconstruction. That's sort of the high level intuition about how that's supposed to work. And so this is kind of talks about the way of doing this both ways, right? I think this is what you mentioned. I think if I understood what you were highlighting, Jeff, you mentioned the bottom path. So if I'm in a familiar environment and I see a landmark, you have an observed landmark that can activate. This is so observed sensory perception that will activate a place cell state, which will give me a noisy grid state, which will then infer a grid state for me. Alternatively, I moved to a known position and I don't see any landmarks, etc. I get a known grid state, which will give me a place cell prediction, which will infer a landmark. So it's very like this entire scaffold is very much they both predict each other all the time the same. Like no matter where you enter this scaffold, like the one predicts the other constantly. So episodic and sequence memory. This is the sequence memory bit. And so what they did was.
Can you go back to that with that little? They had that little hexagon. So what is that?
That's what I'm trying to remember. What was the sequence memory? What was the little hexagon?
I mean, it's implying that somehow the hippocampus can drive movement.
I think this is the sequence memory. I think this is supposed to mean is like if you're displaced, then the next place is going to be this, and that place will tell you where to move in the grid scaffold.
But I'm sorry, the hippocampus is driving that thing. I'm confused what that. It's like a second. Go ahead.
Sequence of features. So you're taking the previous feature, and that's giving you what would be at position t+1. So whatever you see now, I see at t equals 0, and then I take that, and then it should predict what I see at t equals 1, and then t equals 2. And
But
so
I'm
it's
sorry,
conditioned
Bobby.
on the feature they use.
One, two,
I'm just
three,
saying,
4, like over here.
a sequence of features. So the hippocampus, I have my place cells there. And are you saying I then observe a new feature? And I still don't
Yeah,
understand what that.
so what you're seeing at t equals 1 depend on what you saw t equals 0. And you're just basically deriving that condition from what you're seeing in the hippocampus or the place cells. So
How
the output of a
does it?
place cell that's how they learn it, I think. They learn the sequence first from, so what's in V is coming from what you saw before in the hippocampus at the previous time step. And
Like this is like a very artificial mechanism of this computational model of how they do a sequence so that your place predicts your vector movement that will then move you. Like this is completely computationally artificial here.
Yeah, I'm still struggling. It seems to me like if you want to memorize a sequence, it's really, as we know, it's very easy to do that in any set of, sparse representations, a sequence of sparse representation. You can remember that as a high order sequence. And just the idea that you have this somehow the hippocampus is driving or learning, training what movements you should make seems odd to me. I mean, on the one hand, we want to be able to say, yeah, if I see feature A and I see feature B, I might want to know what my movement vector is to get between them.
yeah,
But I'm failing to see how this diagram represents that. It seems just odd to me.
I agree. This could entirely happen inside the hippocampus by just connections between just like an HTM, you can have
Okay, all right,
a
right,
sequence there. But I think they wanted
right.
to learn that and involve the grid cells. Yeah.
Okay. Well, that's the first thing in this diagram. That little hexagon says that doesn't strike me as right. So maybe, as you said, Tristan, it's just a way that they did the simulation or something.
Well, yeah, we kind of left the this is kind of the last one that can match and overlap. Like the reason I became interested in vector hash because I was curious if we can use the scaffold, for example, to represent our metric space inside a cortical column. And sensory inputs are simply the features that we're getting, and then the velocity, these are the movement vectors that we'll be getting. Like that would be interesting approach to me. But as we're moving forward, we're gonna get more and more divergent model from what I think we are and what we're willing to accept. So, but I'll just kind of drive through of like, and here's a neat thing, but we're gonna start diverging quite rapidly from what I think we'll find acceptable. Let's just put it that way. Um, so in this one is with this model they were able to learn like really long sequences because they were essentially moving themselves based on this stabilized place cell output from the again we have a stabilized scaffold dynamics then this place cell would predict the next movement vector and then the movement vector will move the grid which would
So
predict the place cells.
they're interested in the sequences here because I think you earlier said episodic memory. So episodic memory is remembering the exact order in which you did things over time and the time it occurred.
Yeah.
So
And so
that's the
this is how they
motivation.
crashed it here.
This is the motivation for doing that here, looking
Yeah.
at this mechanism.
Because what the next thing they're gonna do is they're gonna attach sensory inputs and try to recall things from where they were into place.
Yeah, right.
And so that's the next thing, attaching sensor input, and they're showing, hey, we can recover. You can see the recall in the original. And then lastly, they went all the way down to building a memory palace. And so in essence, what this shows is you have sort of this sensory recovery if you walk through a space, and so this scaffold gives you like really good sensory recall that sort of resembles your original input. But what they then did that they introduced another network of association between like items and the sensory input, like literally like a memory palace. Build a memory palace. So you have things you have places that you observe in a space. You define a path through it. You place items along the path, and then you want to be able to recall the items. And again, this is we're now getting very artificial here, but just highlighting it as a mechanism of what I think is worth highlighting here is the sensory recall out of this sensory scaffold goes very poorly. Like you can see, it's deteriorating severely after very long sequences. But this memory palace, this little dynamic of when I'm gonna attach items to these sensory inputs as a separate network, it gets recalled actually pretty well. So there's some sort of stability going on here if you like one space removed from this thing. But other
Hey,
than this high level thing, there's I don't have much else there.
I always felt this is again. I haven't read this paper, so I apologize for that. But the memory palace is almost it's exactly our mechanism in some sense. I mean, you're just trying to remember. Normally, we remember where things are in the world. Like I'm looking around my house right now, and I see all these things, and I know where they are. Right, and if I go someplace, I know what I'll find there. And the memory palace is basically imagining that there were things that you wouldn't typically associate with some location. Um, but it's like if I want to memorize a set of cards, it's literally as if I say, "Oh, I left the two of spades on my kitchen counter. I left the four of diamonds by the front door." It's not a new mechanism. It's the same mechanism. The way I remember where the chair is the way I remember where that card is. It's really no different. And it was I'm not sure what they're implying here, but you don't have to do it in the episodic serial way. When the memory palace doesn't require you walk through in the exact same pattern, unless you're trying to remember the sequence of events. Like if you're trying to remember the sequence of events, then you have to imagine the sequence of movement through your house. But if I just want to remember a series of a set of objects, I don't have to do it in that way. I could go randomly through my house. Um, so there's two separate things here. One is associating, unusual objects with locations, which is just the standard mechanism. And then there's a separate one, which is say, oh, is there a defined path that I can learn or know that allows me to go through those locations in a regular way. So there's really not a lot. The whole memory palace thing is really just falls out of the basic theory. I guess I'm saying it's not like something else to explain.
And so yeah, so this is as far as they went. What I would say is this, yeah, right here. This is probably the up here in the upper right. This is probably the interesting bit for because if I squint at it, like I could see like maybe we could implement a learning module like that.
Yeah. So
Like
so it's are we at the end of this one?
We are at the end of this one. This is the paper for further reading if anybody's interested.
Can we just go back to that one? Oh, no, that one
This
right
one?
there. Nope. The three layers. Simple picture. The three layers.
Oh, this.
Yes, that one.
Okay.
Okay. This directly correlates to our learning modules, and both with one major difference, and this is a difference we just talked about recently. At least I read about it recently. I think we talked about it recently. This is an inverted version of our cortical column. Grid cells are location layers on the bottom, layer 6A. What's labeled here, hippocampus, those play cells, that would be in layer 4. And we have sensory input coming into it. The big difference, if I wish I had a picture of our cortical column next to this, but you'll have to imagine it, right? The big difference, the way we envisioned this working in our original papers and how we talked about it until very, very recently, is we assume that when a sensory input comes into layer 4, so the hippocampus in this place is represented by layer 4, sensory input comes in there, we assumed that we would randomly pick a cell in each mini column to represent the location. That is the association between the location between what's labeled here as the grid cells and the hippocampal cells. We assumed that was randomly chosen and then learned. And then you could associate the sensory input with the random association in layer 4. The big difference here is that the association between the grid cells and the hippocampus, that is the association between layer 6A and layer 4, are not randomly chosen. It's fixed, right? You've said it yourself many times. It's a fixed relationship. So if I know if I pick a grid cell location, say there's several grid cell modules, and I pick a location in each of them, that will, without any learning, determine what place cells become active. And if I specify a set of place cells, that would be determined without any learning which grid cells should become active. That is a change from what we assumed before, where we assumed that the connection between the grid cells and the layer 4, the place cells, was learned. And now here they're saying it's fixed, but after that, it's all the same. But I think that's a major change. It's not like a simple change. What it suggests is if a sensory input can specify a set of place cells, it would basically say, based on some sensory input, if I had the same sensory input, I would always invoke the same grid cell modules in the same location. It's not it's always going to do that. And so it almost feels like, how do I describe this? This mapping between grid cells and place cells, it's sort of like this, as you said, fixed location. It sort of suggests that I could have multiple sensory inputs which might map onto that particular place cell mapping. And so again, we could have, it almost suggests it's like a class of objects as opposed to a very specific object. That's the best way I can phrase it.
What if we thought of it as both the grid and the pink box being in the deep layers? And so basically, like the unique location representation on that object is in layer 6, and then the learned connections are between layer 6 and sensory is not the layer 4 input, but whatever gets activated in layer 4.
Well, except in that case, we do know the sensory input's going into layer 4. So,
well, yeah, it activates something in layer 4.
But
then
that would add another layer of cells to this picture, because if we put the hippocampus place cells in the lower layers, then the layer 4 cells aren't in the picture anymore. It could be. I'm not sure what the advantage of that is. My point is that
I mean that the green one is basically the layer 4 cells.
Well, I don't think that's how is that what they're saying here?
I mean, it looked like in the first picture, it looked like those were like object ID STRs, for example.
Well, I don't think that really makes sense. Well, it does make sense. I don't know. I don't know, Vivian. It could be.
Yeah. I have a few comments as well, I guess. Could you go back to the 3-layer, Tristan? Well, I guess this model, first of all, is in kind of line with like the other models that we looked at, Jeff, a couple weeks ago about grid to place cell
Same lab,
models.
same people.
Yeah.
No, I mean like the Abbott, like 2012 Abbott paper, for example.
I don't remember. What was it? Was it that the one that said that this was a fixed relationship between the hippocampus place cells and the grid cells?
Remember we were discussing the equation. It's like the equation that goes from grid cell to place cell.
I remember discussing. I don't remember what paper it was or what we.
Yeah, I mean, yeah. Or I can
Okay.
open the Excalibur, whatever.
No, anyway, I was saying it. This is what we talked about a few weeks ago. That this. But I'm assuming, unlike what Vivian just said, I was assuming that what's labeled here as hippocampus is layer 4, but maybe not.
Okay. Like I was talking. I mean, like I was talking about this and what we talked when you presented, and I guess I guess what I want to see is some other model for kind of hippocampus that's not just based on grid cells, because we know that a lot of things can give rise to place fields, even if there's like complete inactivation of the grid cells, like even if that grid cells go away, we can still see a place cell activity, and we know that there's other cells that influence like place cell activity. So this is a very
Right.
simple,
Well, but this figure explains that. It says basically sensory input doesn't go to grid cells. It goes to the hippocampus, the place cells, right? So once you've learned something, your sensory input would map on the set of place cells.
Yeah. I mean,
you just wouldn't get path integration.
Yeah.
Is that? I'm sorry, I don't want to speak over your but I mean, Vivian, Jose.
No, I guess I'm not really like satisfied with these like grid to place cell networks. Like, forget about the sensory layer. Like, I know that when we have these models, they can have really desirable properties, and we found it surprising that the layers between the two can be random and fixed back in the 2012, not from the field, like that. Those can be fixed and still generate reasonable place cells, but um anyways, I guess that's just my desire from like
What's your desire? Explicitly.
the that the place field, how the place fields are generated is not just dependent on the grid cell modules, but other things that exist biologically, like border cells or head direction or whatever.
Well, presumably border cells and head direction, border cells would be part of the sensory input of some sort, right?
Right.
Sensory doesn't mean it's coming right from the retina. It could be saying, "Oh, I see a window at some distance from me," right? I'm inferring there's a distant object at some pose to my body. That would be that to me sensory, but it's not like it's coming from the retina. It's often it could be, but most of the time it's like an object you're observing. If a rat goes into an environment and looks at some distant objects, those are its sensory inputs.
Yeah.
And they can map to the hippocampus.
Yeah, and the other thing is that like what you said about the sense, if kind of representing object classes instead of a specific object, like I don't think this allows for kind of slightly different representations of objects depending on context. And I think that the kind of flexible representation is something that we want. So if you go back,
Okay, fine. I don't want to leave that thought. If you're going into another thought, I'm gonna stay there for a second.
okay, Chris, can you go to the memory palace one where we're forgetting like the like I guess I want to know people's opinion about figure D, where we're kind of the sensory recall after 2000 or 3600 steps. Like, when I feel like for me, this doesn't really represent how we forget or kind of like, I don't think like all the whole input becomes a little bit fuzzy like this. Like, I feel like we forget like certain.
I don't know what that figure means. What's going on in the figure?
Well, this looks like for me, it looks like we're just continuously adding more Gaussian noise until everything looks kind of blurry.
But what is the sequence like referred to? And I'm confused.
I'm guessing like I thought that this was like after.
So if you like try to memorize a bunch of things in a memory palace, you have a single sequence you're going through your space in, and so this is my understanding is the 2000 steps is 2000 steps through your memory palace. This is what the recall of the sensory input is versus the blue item on the bottom, which is the thing that got kind of placed on there. Like it's just the farther down you go the memory palace, the worse your sensory recall.
But that's weird. I wouldn't think that's right. I mean, if
Yeah,
I'm
as soon as
just
I said it, I think I'm wrong. So never mind.
well, no, but maybe that's what they're saying. The whole point is if I'm imagining myself going through my house, I'm just imagining it, maybe. I don't get it doesn't get fuzzy. It's not like I get lost where I am, if I'm doing path integration in a room and it's dark, yeah, I immediately start getting noise because but as soon as I have some sort of feedback as to where I am, then
Yeah.
bingo, I'm there. So the point of
Yeah.
this is you wouldn't put the two of hearts. You don't replace the chair in the house with the two of hearts. You remember there's the chair and then it's associated with the two of hearts. It's like so when I go in my living room, as I walk in the living room, I'm like, oh, I haven't been here a while. I don't remember what I'm going to see. No, it's not that way at all. Anyway, so I didn't understand that interpretation of the figure D.
Yeah. Well, I guess I for me, it doesn't represent like what human memory decay would look like.
Well, is this
yeah.
human memory decay or is this noise due to path integration? That's what I
I
said.
guess that's a.
When
Okay, I see.
it didn't say trial 800 or trial 30, it said sequence length. So to me, that would represent path integration decay or sort of capacity limit of the network or something like that.
The other
Um,
thing it might be is the capacity decay. If you try to
right,
remember 800 steps versus 2000 steps versus
right. A
3600
limit,
I
a
steps.
see.
capacity limit of the network.
Yeah, I think that's what
Right.
it means.
Okay. But does that mean that's weird? I still don't get it. I didn't put much weight in that figure.
Okay.
Yeah, I mean,
I don't know
I think what you're saying, OJ, is maybe that like it doesn't feel like we remember the two of hearts more blurry if we try and do a longer memory palace, but it's more like either you remember it or you don't remember what was at that location.
Yeah,
And
or
so
maybe you just remember the two or the red, like, but it's like a whole element missing that, but it's not like a whole decay of the whole thing kind of in a uniform manner.
yeah,
I'm pointing this out because this kind of diagram is very common and I because this was a PhD, like autoencoders, diffusion models, like you add some noise, you denoise them, this kind of thing. So like I'm wondering, I saw that in some figures, they did some comparison to autoencoder, like I'm assuming it's some kind of denoising autoencoder. Um, this is how your, like stable diffusion things in some sense kind of works. I'm just like, what's different? Like what makes it, I guess, kind of, I mean, I know the architecture is different, whatever, like there's some fixed weights, there's some backprop. I get that part, but yeah, like, yeah, like,
I'd say that this is more biologically plausible. It's using local dynamics to recover, just like Hopfield networks and things like that.
right.
But also you
Right.
add to it the movement component, and you'll be able to recover not only uh, patterns or like separate patterns that are not dependent on each other, but you add to it temporal dimension, or you could add to it spatial dimension if you use grid cells. So
I guess the
uh,
difference here to what we would do in Monty is that here they're trying to associate that entire image, like all the pixel values, versus in Monty, the model of that bird or the birdhouse, whatever that is, chime, it would live
I think
somewhere else, and you just associate the SDR for it, the object ID, with the location, which requires a lot less capacity.
Right, right. So some of these neural network simulations, I have no idea what they're doing in this paper. There's so many of them. They have all these neurons are partial activations. So this image, it maybe thousands of neurons that have some partial activation. That's not what happens in the brain. We have sparse representations, and those sparse representations they can auto-associatively learn each other. So they lock in the same way the grid cells do, right? They lock in. You don't get these in-between representations. Once you learn something, either you're A or B, there's no in-between A and B. It's going to lock into one or lock in the other. And the neural dynamics do that. So, but many of these sort of early neural network simulations, and maybe that's what's going on in figure D here, is they're not doing that. They're just having a whole bunch of neurons of partial activations, and you're trying to learn these refined weights between all these different patterns. They ain't the way the brain works. It doesn't work like that at all. So it's like it's more like the grid cells. You're going to lock into A or B or C or D, but you don't lock in. You don't have these. It's never happy in these in between states. So it's either going to say this is the bird again, or this pattern A again. It's not really a bird, but this pattern A again, or it's going to lock into another learned pattern. But it's not going to give you these sort of fuzzy in betweens.
but are you always able to extract exactly the same SDR? I mean,
Pretty
I think
much. It's very hard not to. I don't know. Maybe you can explain, Rami, but it's hard not to if you've learned it. If the SDR cells and the SDR are associated with each other, then it's self-reinforcing. In fact, you could just have imagine I have an SDR and it says 5,000 cells and 100 are active, right? Even if I just picked 50 accurately and 50 inaccurately, it would lock in on the original 100 again. It would, no question about it. It would just say, there's only, if I learned it, I have to learn it, of course. But if I've learned it, it will do that. My point is, you don't have to see the whole pattern. You can just see a small subset of the pattern, and that's sufficient to uniquely identify the pattern and have it fill in everywhere. We rely on this for our voting mechanism between columns. It's sort of a part and parcel to the whole all neurons work everywhere. It's part of sparse SDR theory.
But as you start to hit the capacity limit of what you can store, there will start to be some interference between the patterns that you've learned.
Right. Well, you'll start. But I'm not sure that's what they're showing here, because to me it looks to me like they're showing these sort of grayscale activations, or these variable activations. And so much of neural network theory is based on these kind of non-SDR stuff. But you're right. Obviously, there's a capacity to how much you can store, and it's not hard to actually calculate that capacity. We've done it. And if you start too many things, then what the failure when you start representing too many things is that the general failure mode. If I have an associative memory between SDRs, and I'm trying to represent too many. What happens is that individual items that were initially individually represented get combined into a single entity. It's not like you just get completely confused. It's more like you lose the ability to differentiate between two patterns. They become one pattern. It's like, okay, and it's almost like a class. It's like, okay, these patterns are all represented the same way.
Does our SDR, does our representation, can they ever update as we learn more? So as in, for example, let's say I learned a tomato, and I thought it was a vegetable, but then later, I learned that it's actually a type of fruit. Like, does that kind of, but nothing about the tomato has actually changed.
So that's an association between one SDR and another SDR, right?
Okay.
It's between tomato and something else. So a particular SDR can be associated with many, many other SDRs accurately and simply, right? And so I could say it's a vegetable and a fruit. I could say it's something I grow in my garden. I could say it's my favorite thing to hit my kids with. I mean, whatever you want. You can associate a lot of things. It doesn't change the SDR for tomato. It's the memory. There's two forms of memory in a basic SDR circuit. You got the one form of memory is you're associating one SDR with another SDR, and you can do that a lot. There's a lot of those you can do. Many, many, many. And then you also want the SDR itself to be self-reinforcing. So that would be between the cells in a particular layer, in the same column, for example. And that's a different form of memory. That's just saying, hey, this is one thing, we're going to remember it. And it's different than other things. And if you chose the initial cells randomly, it's very easy to do this. If you don't choose them randomly, then you have to come up with a mechanism.
Okay, sounds good.
Can we go back to the simple picture of the three layers? I just want to remember everyone. That was the simple one you read there. There it goes. Just to remind everyone, these details may not matter too much for Monty. Monty, the whole idea of sensorimotor learning is you have locations associated with observations, and we have to say there's an object at some pose at some location on another object. That's pretty much it. And we need path integration to infer and learn correctly, because we're doing it sensorimotor learning and sensorimotor inference. But beyond that, beyond saying, oh, there's an observed object at some pose relative to, at some location, all these other details are nice, but they're not, it doesn't really change the big picture, right? The big picture is the same in all these scenarios. It's just we're representing objects by a series of space, and we're associating essentially childs or features at poses in that space. And none of these details change any of that, as far as I can see. So it feels like there might be some important lessons in here, but if we got it wrong, I'm not sure it would really make too much of a difference. I don't know, but I just want to remember none of this changes Monty in any fundamental sense at all. Does anyone disagree with that or feel otherwise or question that?
I think the biggest thing is a fixed thing. I think that's still the most surprising. The fixed
It is
relationship
surprising,
between
but the question, Jose, does it change fundamentally how we would build Monty?
uh,
I mean, yeah,
yeah.
that's why I said maybe about what if we conceptually think about the grid and place cells to be in the lower layers and then the features in layer 4, because I mean, I feel like conceptually it's still the same thing. We want to have a unique location representation in a place cell, and we want to associate that with a feature, and we want to learn that association. And so whether the way we get the unique location representation is by a fixed relationship between grid and place cells or a learned one doesn't really matter too much for that.
Right.
Yeah.
It's the same with grid cells, right? We deduce there has to be a reference frame for every object. We deduce that. And it's like a fact. It has to exist. And so then we say, well, how do the neurons do this? Oh, there's going to be some cell representation. Oh, look, there's these grid cells. Oh, look at all the clever mechanisms that grid cells work. But as long as our system has unique representations for location and can do path integration, it's pretty much all we care about. We're never going to model grid cells in Monty. We're not going to model the neural dynamics of, continuous attractors and all the stuff that neural systems have to do. It's not that important. It's not important at all. It's important to get the principles right. And so I just remembering that the principles that we just stated, that you've got locations and features that beyond that, it's all details and maybe not matter. When we're going through papers like this, I'm asking myself, does this change in my understanding of the relationship between place cells and grid cells? Does that change in how I thought about it make a difference? That's what I'm asking constantly. Does it make a difference? Is there some new great property that comes out of this that we didn't anticipate, or some new important feature that, I don't know. At the moment I can't figure out what it is.
I
But
have one.
that's what I look at it. You have one, Tristan.
Yeah, so here's why I found this entire mechanism interesting a long time ago, which was when we get a feature into a learning module, we generate so many hypotheses about everything, and then we narrow that down. What I found compelling about this mechanism is there is a learned mapping from you get an input, it hits a place cell, a grid cell, and you immediately get a reference frame. And so I was looking, I'm not saying this solves it, but that's what I thought. This is why
But
I
but
explored
remember,
this.
Would you
but
get
remember
that? I
this.
mean, if you just do the sensory input and the sensory input isn't unique to one location, wouldn't it still activate many locations in the place cell layer?
Right,
Yeah.
it would.
And we still need to learn the hippocampus and sensory. So we still need to learn the association. So when we're learning, we're still going to have a lot of hypotheses, right?
I think Monty has a general issue of it learns a lot, and and it's not efficient in that regard. And there's multiple ways of there's ways of getting around it, but I don't think this mechanism on its own tells you that. Um, uh, a better analogy would be when an animal enters, and I mentioned this a couple weeks ago, when an animal enters an environment, what environmental clues does it use to determine its environment? It doesn't use local environmental clues. It's known that the animal uses distant environmental clues, big objects far away. These are more, and you can understand these are much more likely to tell you where you are and where you are in that thing than some local clue, like a big feature over to the northwest and another big feature over to the southeast. Those could uniquely identify the room I'm in. Whereas the local doorknob or the little feature on the floor in the rat's cage, that probably won't do a good job at that. So one way that, when we do our explorations in Monty and we have a sensor come in, we don't have an equivalent of the far large feature extractor. We just we're like crawling around on the floor, like looking for little crumbs on the floor, trying to figure out where these crumbs are arranged relative to the last time I was in this room, type of thing. So if my lesson would be, if I wanted to try to make Monty more efficient, I would look for ways of maybe thinking about following nature's clue. Like the rat doesn't use local clues. The rat somehow figures out distant large clues. And is there can we do that same type of thing? Um, um, and I have lots of thoughts about that, but that's to me how I would address the problems you're talking about, Tristan.
Okay. All right. So just as a reminder, why do we talk about vector hash at all? I just want to tie it back to this model here is what I saw Joe testing in rats, the relationship between grid cells, place cells, head direction cells, etc. So there might be evidence for this sort of thing happening in rats and to
Right,
some
right.
level of approximation. That
Well,
was
again,
so again.
yeah, it's good. It's a good. I'm really glad you bring up these papers. This is actually very really useful, Tristan.
Yeah,
Going
thanks
through
for
this.
sharing that.
But I do want to remind myself, none of this is really basic what we already have. This picture right here, this is what we've done.
Yeah, which is literally why I was like, oh, is this an alternative code for in the guts of the learning module? Like literally, I
Right.
wasn't like trying to come up with another theory. I was like, no, how do
I
I
got it.
how do I get myself a metric space? And here we have XYZ, and I'm like, can this scaffold be the metric space? Is
I'm
sort
just
of
trying
where
to
I came
figure
at
out.
it from.
I'm trying to figure out could the fixed black arrows here, the fact that they're fixed, make a difference in a fundamental difference that we would want to take advantage of in our models because we don't have that right now.
Yep.
And I can't put my finger on it yet. I can't say that. It may not. It could be just the way nature did it. Not everything in nature makes a hell of a lot of sense. Just or it's something wrong with it, but it doesn't make a difference.
Yeah, and I guess in a
I mean, it's
sense,
amazing.
what we have in Monty right now is basically the fixed version because we have basically in Monty grid and hippocampus is just an XYZ metric space, and we don't learn that metric space. We just initialize it and then do math on it with the B movement vector input.
We don't have an equivalent of grid cells, but maybe you could say any particular XYZ coordinate is a grid cell. It's like,
Well, it's not a grid
It's not
cell
like
layer;
it's
it's
a
just at the
place cell,
location,
right?
right?
I mean, place cell.
Yeah.
I'm sorry, place cell. Place cell. I meant place cell, right?
Yeah. It's like every XYZ coordinate is a place cell, and then we don't need grid cells because we can do path integration in Euclidean
Right,
space by
right,
doing
right,
math.
right. It's funny. Neurons can't do math, right? They just can't do math. So there's no way to say, oh, what's my XYZ? Here's a bunch of cells that represent XYZ. They can't do that. You can imagine, oh, I have 3 cells that have variable firing rates. Forget it. That doesn't happen. So everything has to be done with some sort of sparse coding. Everything. So nature has come up with a whole bunch of ways of doing what we would do with mathematics. They do with sparse codings. And does it make a huge difference or not? Look, Monty works today without any sparse coding, and so.
I just wish the larger deep learning community would see this and be like, hey, we don't need to learn weights at all. Like literally there's no backprop because the weights are fixed and things still work. It's like, okay, let's take a
Right,
minimalist
right.
and then like try to do something, based on that, I think would kind of move
The
everybody
funny
all
thing about
together. But
the funny thing
yeah,
about the deep learning communities, they're modeling neurons, and they're modeling incorrect neurons. They're modeling point neurons.
right.
But it's funny because I would say they're not neurorealistic, but they're modeling neurons. We don't need to model neurons, but I think we're more neurorealistic because we're capturing the larger principles, which they're missing completely. It's funny, they just skipped down to these low-level details and built the whole system based on these low-level neural networks, but they're missing all the large structural principles of how the brain works.
All right, so we're gonna put this as a thumbnail so that it's like everybody who sees the video and then we'll say like black is fixed weights, and so that like this will be like a so that even if somebody doesn't watch the video, they'll be like, oh, there's fixed weights.
Yeah. Also, in this picture, I assume they're showing 3 little circular loops of the grid cells. I assume they're saying, oh, we're going to assume there's like 3 grid cell modules. Is that right there?
Well, it's not 3 specifically. It's configurable in the actual
Well,
code.
okay, but it's funny because when we did these recent papers trying to figure out how many of these overlapping grid cell modules are there, that's like how many would be a bit, visible to a color column. We came up, number 3 was sort of like the middle number there, right? It could be 2,
Yeah.
it could be 4, but
This
So-
method gets computationally expensive once you get to like 6 or like beyond
right.
5. It gets a lot.
Well, maybe, but the different point is 3 sufficient? Um, It's funny that maybe the neuroscience community is settling on. Okay, 3 seems to be what we're going to think about. That's new. That didn't used to be. It's new in the last, I don't know, 15 years or so.
Alrighty. And so this is the paper for the vector hash thing. And then I had a bonus, but I think we've talked long enough. So
You can't tease us like that.
We don't get the
Yeah.
bonus.
Yeah.
so this is
We were well behaved, Tristan. Why didn't we get a bonus?
Okay.
So this was a bonus. I've mentioned it in the coffee chat a little bit, but Cephalopod camouflage is sensorimotor, something I didn't know. So
It
the
can be
way
hormonal, like in chameleons, or it could be.
well, it's
yeah.
sensorimotor because so this is zoom in skin in the upper right here, and so the way they change both the texture and the colors is they change the sizes of the different chromatophores that they have in their skin.
Yes.
And this picture down here, this is a chromatophore, and all these are motor neurons. Sorry, muscles that are
Yes.
innervated by motor neurons, and a motor neuron will innervate like an area of some of these muscles. And so, well, the bonus here is kind of highlighted here. Retina and inverse retina is what these things do is they see something, and then that travels through their nervous system in a sensory motor way. So they actually activate motor neurons, which actually activates muscles, which changes their camouflage. Then they see it, they see how far off it is from where they want it to be, and then they go through the tighter loop and a tighter loop. So literally, it's a sensory motor cycle for a cephalopod to change its camouflage. I thought that was
Wow, it's
neat.
pretty.
So they
Very
can
cool.
see themselves with their eyes because
Yes.
they're
Yeah.
intruding.
And what they're matching. Yeah.
Yeah. That's
Are
why
you
they're so
sure?
fast in
I
camouflaging.
Yeah,
don't
this
think
was like
they're-
a whole presentation on it. I was just like, this is just
I
a
don't think the
teaser.
cephalopod is thinking about this.
Oh, no, not thinking.
I mean, how do we know that? How do we know it's just not fixed?
There is so I didn't have a slide, but it's they've measured this, and it's essentially it takes like it does it's a diminishing returns of fidelity. So it sees a pattern, then there's like a step of one cycle of a neural loop where it gets closer, and then it gets ever closer to it the longer it's exposed to the pattern. But it is step functions. It's like walking through the spaces of camouflage available to it, and it's an iterative process that they've actually measured. It's not like a reflex response to a specific stimulus. It's actually walking its path towards matching better and better and better over time.
Wow.
Yeah, it was
Octopuses
kind of
are
cool.
That is really cool. That is very cool.
That concludes my presentation for today. Thank you.